This video will be part 2 of changes in chromosome structures. Inversions occur when a segment of a chromosome is flipped into the opposite orientation. There is no change in the amount of DNA here, so often there are no phenotypic consequences. For inversions, there are two types: pericentric, which contain the centromere in the inverted region, and paracentric, which do not contain the centromere in the inverted region. While inversions often do not have phenotypic consequences, under certain conditions they will. These conditions largely have to do with where the break occurred in the chromosome. For example, in the middle of a gene sequence or in repetitive DNA. Also, if the break occurred at a critical position that helps regulate gene expression. About 2% of humans carry an inversion that is large enough to be detected under light microscopy. While these individuals are often unaffected, they may be more likely to produce abnormal gametes. Our last category of structural change in chromosomes are translocations. These occur when a segment of one chromosome becomes attached to another chromosome. When two non-homologous chromosomes exchange segments, this is called reciprocal translocation, and it happens via two mechanisms. These mechanisms are chromosome breakage and DNA repair and non-homologous crossing over. Often, these reciprocal or balanced translocations are simply rearranging DNA and thus don't have a phenotypic effect on the individual that has them. However, the same issues can arise as we saw in inversions. The break point is at a critical spot on the chromosomes. Translocations are often most impactful on the phenotype when they are unbalanced. When this happens, some genetic material is usually duplicated or lost. An example of this is familial Down syndrome. This is caused by a translocation where most of chromosome 21 is attached to chromosome 14. An individual that has this arrangement can be unaffected, but produces a high percentage of abnormal gametes. The schematic for familial Down syndrome is shown here, and you can see that half of the possible gametes a carrier of this translocation produces are lethal. Two would result in normal phenotypes, and one would result in a Down syndrome phenotype. While a person with the familial Down syndrome phenotype actually has 46 chromosomes, the normal number, due to the translocation, they have three copies of most of the genes from chromosome 21. This is an example of a broader type of translocation called a Robertsonian translocation. These types of translocations occur when breaks near the centromeres of acrocentric chromosomes occur. The P arms of these chromosomes are often lost, and the Q arms combine to form a large chromosome. This is a reasonably common rearrangement in humans and occurs in approximately 1 in 900 births.